Hello everyone, welcome back to my channel. So in this video we will talk about serotonin, which is also called 5-HT, that is 5-hydroxytryptamine. So जो serotonin word है that was initially given to a substance which was vasoconstrictor in nature, and it was found in serum when the blood was clot. Okay, and there was another word that was antiramine, which was given to a substance which was smooth muscle contracting, which was found in the enterochromaffin cells which were found in gut mucosa. So eventually both were found to be 5-HT, that was 5-hydroxytryptamine. About 90% of the body's 5-HT is present in enterochromaffin cells of stomach and intestine, and the rest is present in brain and platelet. So now we will discuss about synthesis, storage and destruction of serotonin. So 5-HT chemically it will be 5-amino-ethyl 5-hydroxyindole, and it is derived from amino acid tryptophan, and it is degraded by MAO. What is MAO? It is monoamine oxidase, and some extent it is being degraded by dehydrogenase. So tryptophan will be acted upon by hydroxylase, and it will produce 5-HTP. So 5-HTP, which is 5-hydroxytryptophan, on which decarboxylase will act, and it will produce 5-hydroxytryptamine. And if MAO, that is monoamine oxidase, will act on 5-HTP, it will produce 5-hydroxyindole acetic acid, and if dehydrogenase will act, it will produce 5-hydroxytryptophol. So this decarboxylase is non-specific. It will act both on DOPA as well as 5-HTP. What is 5-HTP? It is 5-hydroxytryptophan, and it will form DA, that is dopamine, and 5-HT respectively. And 5-HT uptake is because of a amine pump, so serotonin transporter, that is SERT. It is a sodium dependent carrier. Okay, it will operate on the membrane of platelet and serotonergic nerves ending, and it is inhibited by SSRI, that is serotonin reuptake inhibitors, and TCAs, that these are antidepressant, these are tricyclic antidepressant. Okay, so platelet don't synthesize 5-HTP, but they acquire it by uptake during the passage to intestinal vessels. Okay, so like catecholamines, 5-HT is also being stored in storage vesicles, and it is uptake by a vesicular monoamine transporter, which is VMA T2, and this uptake is being inhibited by a drug that is known as reserpine, which causes depletion of catecholamines as well. Okay, so now we will discuss about serotonergic receptors, and it was classified initially on the ability to be blocked by dibenzylene, which is phenoxybenzamine, and as well as morphine, into two groups. One was D type and another was M type. What was D type stand for? It was musculotrophic, and M type was neurotropic. But the present system of classification is based on molecular characterization and cloning of receptor cDNAs. Okay, and the subtypes of these families which are being classified, it depends upon the tissue distributions, but certain tissues they express more than one type. Okay, so four families, 5S, T1, T2, T3 and T4, comprising of 14 subtypes, has been so far recognized, but almost few are functionally correlated. Okay, and why we are talking about subtypes? Because the knowledge of subtypes is important, because जो new drug होती हैं which are being developed, they are described in term of 5-HT receptors subtype, agonist or antagonist. Okay, so now we will discuss about receptor. जितने भी रिसेप्टर होते हैं 5-HTP receptors, all are G protein coupled receptor except one, that was 5-HT3, which is a ligand gated cation sodium potassium channel, and because of activation on this receptor it will cause fast depolarization. Okay, So we were talking about that all serotonin receptors are G protein coupled receptors, and they function through decreasing or increasing the cyclic AMP production, which was the cyclic AMP production was decreased by action on 5-HTP receptors and increased by T4, T6, and T7 receptors. Okay, or they act by generating IP3 or DAG, diacylglycerol, as second messengers. We will talk about it later, and we also have talk about it. So 5-HT1 receptors we will discuss specifically now. So six classes, six subtypes, 5-HT B, D, E, and F have been identified, but जो 5-HT1C, initially it was in this category, but now it is 5-H2C. Okay, so all the subtypes, they are GI or G0 type of coupled receptors, and they inhibit adenyl cyclase. And these receptors, they function primarily as autoreceptor in brain, or inhibiting the firing level of 5-HTP neurons, or release 5-HTP from nerve ending. So basically they are inhibiting, and they are inhibiting the firing of 5-HT neuron in the brain. Okay. And another subtype of this receptor, that is 5-HA1, 5-H1A, these are somatodendritic synapses which are present in raphe nucleus of brain. And what does they do? They reduces the firing of raphe nucleus. Okay, and the next center for this is hippocampus. It is also another important site. So anti anxiety drug, that is busprayon, ये एक पार्शियल एगोनिस्ट होता है ऑन दिस रिसेप्टर्स. ओके, सो 5H1 5HTD or 5HT1B, they regulate dopaminergic tone in the substantia nigra, and we will talk about these drug later, but it is sumatriptan, which is an anti-migraine drug, and it is a selective 5HT1D or 5HT1B agonist. Okay, and we should talk about other subtypes as well. So 5HT1P receptor, they will express on neurons of enteric nervous system and mediate their slow depolarization. Okay, so now we will talk about another receptor, which is 5HT2 receptor. So there are three subtypes, and all are GQ coupled receptors, and they activate phospholipase C, and they will produce IP3 and DAG, that is diacylglycerol, as a secondary messenger. 5H2A, it inhibit potassium channels, and it also causes slow depolarization. Alpha methyl 5-HT, it is selective agonist of all the three subtypes of 5-HT2 receptor. Previously, 5-HT2 receptor was put under a category of D-type receptor. Most importantly, they are present at post-junctional receptors, mediating their action, which is direct action like vascular and visceral smooth muscle contraction, platelet aggregation, and neuronal activation in brain. Kitaserin, it blocks these receptors because it is an antagonist. Okay, and choroid plexus, it may regulate CSF formation because choroid plexus expresses large number of 5-HT2C receptors. Okay, 5-HT2C receptor. Now we will discuss about another family of receptor, that is 5-HT3 receptor. These are known as neuronal 5-HT receptors, and they rapidly depolarizes the nerve ending by opening of which channel? Cation channels. And previously they were put under a category of M type, okay, receptor, and they elicit their action through nerve endings. When like through somatic and autonomic nerve endings, they mediate pain, itching, or coronary chemo reflex. And we should know what coronary chemo reflex, we have studied it in physiology, but also coronary chemo reflex is bradycardia, that is fall in BP due to the withdrawal of sympathetic tone, respiratory stimulation, or apnea elicited by stimulation of receptors in the coronary bag. Okay, and now ending in mesenteric plexus, this causes augmentation of peristalsis and emetic reflexes. And the area postrema And nucleus tractus solitarius, we all know that is a chemotactic center, so it causes nausea as well as vomiting. So there is a drug that is ondansetron that is antiemetic, and it goes by blocking these receptors. Now we will talk about 5-HT4 receptor mainly, and also about another receptors. So 5-HT4 receptor, it couples to GS protein, and it activates adenyl cyclase, which has been demonstrated in mucosa, plexus and smooth muscle of gut, and it probably involved in augmenting the peristalsis intestinal secretion located in brain, especially hippocampus and colliculi, which it causes slow depolarization because decreases the potassium conductance. Okay, and there are some drugs which are selective 5-HT4 receptors agonist, and recently cloned 5-HT5, T6 and T7 receptor are closely very related to 5-HT4 receptors, and these are mainly located in specific area, but their function only is not known. Okay, so that's all for in this video. Please subscribe and hit the bell icon.